All right, brings us to the final speaker for the first session this morning is Julie Lucifora, and we're going to change this a little bit and look at the potential of these drugs to target hepatitis delta, since they'll be targeting the hepatitis B surface antigen as well. Thank you very much, and the stage is yours.
Thank you very much for the invitation. So, how do I? Oh, sorry. Okay. So I'll start with a quick introduction, just to remind you that chronic HBV and HDV infection leads to the most severe form of viral hepatitis. And chronic HBV and HDV carriers, they display increased pathogenesis compared to chronic HBV carrier, with higher risk of severe hepatitis, faster progression to cirrhosis, and higher risk of hepatocellular carcinoma development. And therapies for chronic HBV and HDV carriers are actually limited. So as you know, the efficacy of pegylated interferon alpha-based therapy is low, and patients experience many side effects. And bulevirtide, I never know, that has been approved recently in Europe, even though it was a very big breakthrough into the management of chronic HDV infection, requires daily injections, and the treatment duration has not been established yet, so but it will probably long term. And bulevirtide does not target HDV replication. So I believe that we need to continue our efforts to develop new antiviral strategies, additional one that will target HDV. And today I've been asked to talk about the potential of antisense molecule to target HDV. So I do work on HDV, but not really on these antisense strategies. So as John, I will apologize if I forgot to mention any of any important results. So first, I just want to say that, as you know, HDV is a defective virus, and it requires HBV for secretion and spreading. So theoretically, any strategies that will block HBs antigen production or release should prevent HDV propagation. So, and there are different strategies that are currently in development, such as neutralizing anti-HBs antibodies, NAPs, or the antisense strategies. And so today I'll focus on those one. So to target HDV with antisense molecule, you have two options. So either targeting the HBV RNA, as I just mentioned, or targeting directly the HDV RNA. So I will start with the one targeting HBV RNA, because John already mentioned those strategies. And I want to focus on the results that have been obtained regarding HDV. So GNG 3989, that is an siRNA against HBV RNA, is currently in clinical trial for HDV patients. And so the results of the RIFD study have been disclosed at the last ELC meeting by Heiner Wademeyer. And I think the full set of data will be presented later by Olivier Lenz. So I just want to mention here that the siRNA led to robust reduction in both HBs antigen and HDV RNA secretion. And I think it was very interesting. However, 12 out of the 17 patients experienced ALT elevations that required treatment discontinuation. And another siRNA directed against HBV RNA also, so VIR-2218, that has been mentioned also by John before, has been tested in infection models, so either in vitro or in vivo, and Florian Leb will also present the results later. But interestingly, they also show that this siRNA against HBV led to HBs antigen and infectious HDV particles secretion inhibition. And now I'll move to the strategy that will directly target HDV RNA. So there are different HDV RNA species that are produced upon HDV life cycle. And you can see here, so there is the genomic RNA, the antigenomic RNA, the HDV mRNA, and additionally, the genomic RNA can be associated to HDV protein to form the so-called ribonucleoprotein that enters and leaves the hepatocytes. And due to the particular features of this RNA, I think there are possible limitations to siRNA strategies. So the folding of the HDV genome and antigenome into N-branch rod-like structure might limit the access of the siRNA. The association of the HDV protein to the double-stranded HDV RNA might also limit the access of the siRNA. And finally, the nuclear localization of the antigenome and part of the HDV genome might also limit the effect of the siRNA. Because as John mentioned earlier, so this is the mechanism of siRNA activity, and you can see that most of it takes place, all of it takes place within the cytoplasm. And when you look at a schematic representation of the HDV replication cycle, you can see that most of the RNA are within the nucleus. So theoretically, if you use siRNA and taking into account all the limitations I mentioned before, you might only target the HDV mRNA within the cytoplasm and block the S protein production. And this was actually nicely shown by John Taylor in a paper published in 2003, where they designed different siRNA along the whole HDV genome. So some targeting the mRNA and some the genomic and antigenomic RNA. And they could see that those targeting the mRNA were very efficient to block the protein production, but those targeting the genomic or antigenomic part were resistant. And they additionally showed that this resistance was neither due to the structure of the RNA, nor the conformation, or the presence of the HDV protein. And they hypothesized that the resistance of the genomic and antigenomic RNA might be due to the nuclear localization. But interestingly, as you can see here, even though those siRNA are only targeting mRNA, you see a strong decrease of the antigenomic RNA. And this is because when you look again at the replication cycle, the small HDV protein is actually essential for the mRNA and the genomic RNA synthesis. So if you block the S production by targeting the HDV mRNA within the cytoplasm, you will potentially abrogate the whole HDV replication cycle. So Arbutus developed an siRNA against HDV, and they used lipid nanoparticles for delivery. And they tested the siRNA in a HDV-producing cell line. They confirmed that the siRNA was mostly efficient against HDV RNA within the cytoplasm and not so much for those in the nucleus. But, however, when they used that to treat the producing cells, they could show a durable reduction of both negative and positive strands of HDV RNA, suggesting that targeting the cytoplasmic HDV mRNA is sufficient to abrogate HDV replication. But this needs to be confirmed with infection model. So I gathered some results we obtained in the lab. We use a combination of two siRNA targeting the HDV mRNA that were transfected in differentiated HH7.5 and TCP cells, differentiated heporrhage cells, or primary human hepatocytes. And cells were infected with HDV particles, and we looked at HDV replication, so looking at total level of HDV RNA, and you can see that the siRNA are very efficient to reduce the total HDV RNA, suggesting again that targeting only the mRNA might be sufficient to abrogate HDV replication. And this needs to be confirmed in vivo now. And I found one experiment from Arbutus also. So they used HPV-HDV co-infected mice, and they treated them either with the siRNA against HDV or with another siRNA directed against HBV, so ARB1714. And you can see here HDV viremia, and you see that when mice were treated with the siRNA directed against HBV, you can see a decrease of HDV viremia. However, the effect on HDV viremia with the siRNA directly targeting HDV was quite limited. Yeah. And so you've heard also that there are these ASO that are developed currently for HBV, and I really think we should try to develop them for HDV too, because this might actually help to target the nuclear HDV RNA, contrary to the siRNA. And finally, I just want to mention that HDV replication cycle mostly relies on host factors, and some of them have been identified by siRNA screened done by Eloi Verrier. And we might think about targeting this host factor, but as you can see, this is quite important pathway. So we have to think about toxicity regarding such strategies. So I just add this third option here with a question mark. And so I want to conclude to say that I think that antisense strategies are promising antiviral option for chronic hepatitis D infection, but I think that we should further investigate ASO targeting HDV RNA, and that would be nice to combine actually molecule that would target both HBV and HDV RNA, and maybe think about targeting HDV-related host factor. And that's it for me. Thank you.
Thank you very much. Oh, okay. There is, wait.
This is a very quick question regarding, because the sequence of HDV is so that's not so conserved. How do you consider siRNA? I mean, can cover different genotypes, or even within genotype 1, there's not much, not very highly conserved region can be found.
Yeah, it's so sure. I don't know exactly the detail, but this is something that we have to take into account for designing the siRNA for sure.
Okay, thank you.
Yes, this might be actually a practical issue problem, although there are sequencing regions that are more highly conserved, but of course it's an RNA virus. Maybe Julie, so if we think about now the most advanced RNA interference strategy targeting as the envelope of HBV, so your opinion maybe, can you tell us a little bit more what you think, what is going to happen to HDV in the hepatocytes? I think it, on the long term, we could also, let's say, maybe it's going to die at one point, the cells, is going to drop out the stability, so the survival capacity of the hepatocytes, of the HDV monoinfection status. Can you What is your opinion about?
I think it should decrease at least to a very low level because, I mean, the L protein will be produced, so that will inhibit the replication. So you will have a really low amount of HDV if you block the secretion, I would say. However, you also show that HDV can be in the liver of the mice very long. So I don't know. I don't know if we can completely cure the cells.
Just a quick question, maybe I missed it, but if you're knocking down the HDV RNA directly in the cytoplasm, do you continue to get this cell division spread of delta? Can you prevent that with the siRNA targeting delta?
I think the data
Don't know. Not yet available.
I mean, of course, there is the issue if the survival, the capacity to survive for HDV through cell division, that makes everything a little bit more difficult. It might promote the, let's say, the survival in general within the hepatocytes for longer time. But at one point, still, I think it's a good potential strategy to consider that it's breaking down. If you don't have the delta antigen anymore acting on the genome alone probably is also not able really to maybe to persist that long. But this is a question that should be really experimentally addressed. I don't think there are much data in this direction. Massimo.
Julie, very nice presentation. I like it a lot, the reasoning. Just can you elaborate a little bit more on why an antisense oligonucleotide against delta should have an advantage in targeting the non-mRNA, the other isoform, the genomic and the antigenomic delta RNA as compared to the SI? What is the molecular basis for this advantage eventually?
So according to John Taylor's paper, the resistance of antigenomic and genomic RNA to the siRNA was not due to the structure, conformation, or association to HDV protein. And they really think that it was nuclear localization that was the problem. And so I think, but I'm not a specialist, that ASO can target nuclear protein. So I think we should really try for HDV because even though targeting mRNA might be sufficient to block the replication, if you can target also the nuclear one, that would be maybe more efficient. Yeah.
I'm curious about actually the binding site. So according to John Casey's study, so there are about 2 to 6 delta antigen molecules binding to the HDV RNA genome. So therefore my question is there a space that delta antigen not binding on the genome that can be available for the your siRNA binding?
There is space for the polymerase to do the replication and so on. So I guess there should be, I don't know.
Just a real quick fact to add to this conversation. The primary site of cellular function of human RNase-H1 is in the mitochondria and in the nucleus, where it is involved in genomic maintenance. So there's plenty of RNase-H1 activity in the nucleus, which would provide the ability of that arm of the mechanism of the antisenses to work in the nucleus.
Yeah.
Okay, Stefan?
Maybe also thanks, Adam, for that suggestion. We've not done that experiment. You can repeat the John Taylor stuff with the messenger RNA targeting siRNAs, but we have not looked, or I think nobody has looked, whether that may affect cell division-mediated spread, which would be good. One point, I mean, you can't do not, if you use an siRNA, it's still an injectable, yeah? So it's not an oral thing, but I want to stress maybe a little bit what you mentioned with the cellular factors. For example, in the mediator complex, the CDK8 and 19 is a very important, absolute required factor for mRNA synthesis of delta. And there are drugs in development. They are already in patients that you can use in order to look whether direct interference with replication may may be a benefit in addition to bulevirtide to accelerate the degrees of the replication to shorten the therapeutic treatment time. I think that's a very important goal that we should keep in mind in the future. And that would be oral. So these host factors.
So it would
may
be easier
very
than siRNA. That's why I put the question mark for the siRNA strategy against host factor, because small molecule might be better.
Yeah.
Thank you for the discussion. Of course, there is always the safety issues that emerge, but should be explored definitely. If there are not further questions, I think, Julie, thank you very much for this overview. And